concept with Dr. Ben Shabbat of the entourage effect. The idea that there were many compounds, endogenous cannabinoids, some seemingly inactive on their own, but together they make this beautiful music. They create an entourage effect that greatly increases activity.
Rafi basically spoke to me about this entourage idea, and he made a very nice comparison, like politicians, if they go alone to places, nobody pays them any importance. If they go accompanied by perhaps less active components, just the fact that the politician or the minister or the president is accompanied by many people makes it more important. So this is the entourage effect.
I feel that the same applies to the components of the cannabis plant itself. And so many of these are what are called minor cannabinoids, contribute to an overall effect that you cannot produce with a single molecule. This has been a key concept in what the work I've tried to do under his influence.
I was convinced that one should only test pure compounds with one target, whereas it's the combination sometimes of the compounds that can do the trick.
A very serious group of researchers has recently published a paper saying that the endocannabinoid system is involved in essentially all human diseases.
If you combine CB1 receptors and CB2 receptors, they cover most of the organism, at least in mammals.
We are mammals.
We are mammals.
Okay.
But when you talk of mammals, you talk of horses and dogs and mice and rats and rabbits and lions and...
We all share the endocannabinoid system.
I believe so. I believe so. And it has been demonstrated in many species.
Because now suddenly everyone has cannabis in their bodies, or everyone has a cannabinoid in their bodies, so it can't be bad.
These molecules that we all have are so critical from the birth to the death of each of us in health and disease.
He suggested that endocannabinoids in the milk of mothers actually may contribute to the hedonic effects of the milk. And it turned out to be almost true, actually. So he has a great vision. And then I know that he suggested to Itai Bab that he should look at the bone. Why would anybody in the world, actually, without preconception, look at bone development?
רגע. כמובן, דש ודליה. We're going to my lab. We can go through the bigger entrance.
Or to the
small entrance here. My name is Professor Itai Bab. I am the director of the Bone Laboratory at the Hebrew University of Jerusalem.
We're going to see now the cannabinoids in bones?
Yes.
Okay. So you do have a laboratory here.
You've been here. We have discovered the skeletal endocannabinoid system. These are osteoclast cultures. Osteoclasts are cells that break down bone.
Okay.
So she's analyzing the effect of cannabinoids on the number of osteoclasts. This is the growth engine, which is similar in mice and humans.
Mm-hmm.
You see here the brown cells, these are CB1 receptors, these are C receptors in the engine.
We've got a lot of research now going on because now we know there are CB1 and CB2 receptors in those kinds of cells that either help degrade the bone or help rebuild the bone.